So to introduce our next speaker, Martin is really a thought leader in the longevity space. And we've talked a lot about rare disease, but I think it's also really important to think about bigger markets, right? And longevity is potentially the biggest market. It's something that could be helping everyone. It's important to go after big markets because that's what's going to drive the investment to bring the cost down, even to help rare disease or N-of-1 patients. So I'm excited to hear from Martin. He's both the co-founder and the CSO of Gordian.
Thanks, Eric. And yeah, thanks for the invitation. I'll be talking about complex diseases, including diseases of aging, which is where Gordian works. But even if you're not into longevity, you're probably into various diseases, neurodegeneration, heart failure, and so forth, that this applies to. So earlier this year, my dad called me, and he told me that he had cancer. And as you can imagine, I'm a startup founder, I'm hopefully high agency. I immediately went online and started looking up, okay, what does this mean? What can be done? And in this case, he had prostate cancer. And a lot can be done, right? So we were sort of lucky that he got a kind of cancer that we have solutions for. He got surgery, and he seems to be totally fine now. But going through that experience highlights something, a barrier, not just for genetic agency, but for medical agency in general, which is how do we know what to do? Even if we're able to come in with our delivery methods and do something, how do we know what to do when we get there? And so many of us are scientists, maybe all of us. We study disease. We often study disease using cells. Cells are useful because they often divide, and you can do things to them, as we've seen already. And for some diseases, you could argue that the disease lives in the cell. Let's take Huntington's disease. You have a production of something that messes up the cell. That's true in some cases. In other cases, you could argue that the disease lives in the interface between cells. If we think of liver fibrosis or lung fibrosis, you have one cell type that's struggling, dying, and that triggers an immune activation that then in turn triggers fibroblasts to create collagen, and you get scarring. So the disease doesn't really live in any of these individual cells. It lives in their interplay. And you can take that further, right? The organ as a whole is in a certain state. If you have a heart, it's beating. If you have a liver, it's filtering things from the blood. And so I think this brings us to sort of a challenge of like, what are we actually studying when we're trying to figure out what to do? Can we study cells? Can we study organs? Maybe that's feasible. Do we have to study whole humans? Do we have to do trials in order to find the next disease that treats something manifesting all over the body, like, say, metabolic syndromes? This is the challenge that sort of faced us before and when we started Gordian, which is how can we figure out what to do for diseases that are complex and not manifesting purely in the cell? And the question that we asked to try to solve that is, can we ask a single cell about the disease that it is part of and get a meaningful answer? And what we thought might be true is that if you ask the cell questions in the context of disease, meaning in the in vivo system, in a living animal that has developed a disease that you care about, ideally in the same way that humans develop that disease, if everything that you know to be important in the disease and critically, everything that you don't know is important, right? All the things that we haven't discovered yet about how Alzheimer's or heart failure or aging works, if those are all present because you're actually studying cells in the disease context itself, then hopefully you will get better answers. So that's a nice idea. Can we make that work? That was basically the question 7 years ago. There are two questions here. One is, can we ask a cell about the disease that it's part of? And then, is it going to tell us the truth? The core technology platform for Gordian is pooled in vivo screening of AAV therapies that target different genes or combinations of genes, up or down. which this is familiar, right? That's the same thing Dino's doing. We put those in as subsaturating libraries into an animal model. And the animal model here can be whatever you think best represents disease. In osteoarthritis, for example, we find horses that have spontaneously developed OA over a number of years. And we inject them as a subsaturating dose so that you get this mosaic tissue that has individual cells scattered throughout the tissue with a perturbation. You let that happen, and then we use single-cell transcriptomics as basically a phenotypic readout. We measure the expression of every gene, and therefore we can reconstruct every pathway and ask what is going on in this cell. So we've done that quite a lot in different diseases and so forth. Conceptually, the information you get out looks something like this. Imagine this is real data, but anonymized. Imagine every column is a different target or combination of targets. Every row is a curated gene set that's relevant for disease. So in osteoarthritis, one of these will be your IL-1 signaling. One of them will be your collagen production and so forth. And then we see what every possible perturbation does in that environment. We've put a lot of work into the actual computational predictions of disease and interpretation here. And we're getting data on hundreds or thousands of targets in this way. So that answers the first part. Can we actually ask cells inside of the disease, while the disease is happening, what they're going through? And if we do A, B, or C to them, do they move more towards a healthy state? The question is whether they will lie to us. Right? We can do that. We can get data out. You always get data out when you do a screen. But is this data actually predictive of the broader physiological phenotypes that we get in disease? The way that we address that, which is not simple, is to use lots of paired data sets where we take physiological measurements like heart function or fibrotic readouts by histology, whatever is actually relevant for clinical trials. We can get this from patients. When we get patient samples, we can get this from animals when we do our studies. And then we can use those to create predictors and to weigh the different gene sets that we get out of our screen. So that's the core approach. And just to give you a tiny vignette of some of the data we get out here, in this case you're seeing we did a screen in osteoarthritis in horses, as mentioned. Then we took patient explants. So since the lack of a cure for osteoarthritis means that a lot of people get their knees replaced, we can get cartilage from the knees that get replaced, grow that in cold cultures with synovial cells from the knee, and then look at the production of collagen. So that's the x-axis. Each dot is a separate experiment, and the y-axis is what we have trained our computational models to predict from the screen. And we see that there's a fairly good correlation in talking about the production of cartilage just from what we're seeing in our screen. Now, this is still partially a cellular phenotype. So what if we ask about something more complicated, like diastolic function in heart failure? On the left, you have the screen. And we've then done experiments in rats and mice where we use echocardiography to measure the heart function. And we see that we can still predict these kinds of outcomes. We've, of course, taken that further because we're a drug development company and created gene therapies, in this case in osteoarthritis. This is in mice that have spontaneously developed osteoarthritis over 2 years. So again, the most patient-relevant rodent models you can imagine. And some of the targets that we have, they're codenamed OMEN 12 and 13, improve the cartilage of these mice that have developed osteoarthritis. So we can go from doing a screen, probing hundreds of targets, reading out transcriptomes, using the proprietary data sets we have to make predictions around physiology, and then actually find therapies that work in diseases where there is no cures available. So going back to what is the point of that, right? Of course, we're a company, we like making progress, we like making therapies, but if we think about the broader picture of what we are doing here and where we hope to get to, creating genetic agency begins with knowing what to do. And so whereas currently we have agency in what we wear, everyone here chose their clothing, or their spouse did, out of a wide selection, right? You know, we have total abundance of clothing at this point. If I'm invited to like a vampire party or a disco party, I can go on Amazon and have a complete outfit 2 days later. And at that party, I can listen to any music that's been produced in the world. Slight exaggeration, but not much, right? We have such abundance of these different materials. We don't have that yet for what to do therapeutically. When you go to the doctor, like my dad, you have to be lucky in order for there to be something that you can do. If we use mosaic screening to identify targets for most diseases, if we create target abundance, then we can talk about genetic agency, not just for monogenic diseases, but for disease more broadly. So that's what we're working on and making some progress on. And that's something that complements what everyone else here is working on, as Eric was saying, bringing together the ecosystem to solve delivery, solve manufacturing, and then solve what do we actually need to do. Thanks.